Guys, this is Matthew Berry and welcome to my channel. In today's video, we'll be discussing cell cycle control. This is not an exhaustive video, but I do summarize some of the more important concepts in the video. So let's start by looking at the cell cycle. Remember that cells that are not dividing and not growing, they're stuck in the G0 phase or the quiescent phase. For example, some are neurons, some muscle cells. However, cells that are dividing and are growing, they'll be going through these 4 phases of the cell cycle, starting from the G1, where most of the growth takes place, then entering the S phase for replication, where they double the amount of their DNA, then going into the G2 phase for some more growth, and eventually ending with the M phase or cell division phase, where they make daughter cells. And the daughter cells can re-enter the cell cycle, and that's how this cycle keeps going. So the question we ask ourselves is, who runs the cycle? Okay, who pushes for growth? And the answer to that is our cyclins and our cyclin-dependent kinases. So you can see in this picture here that in each phase of the cell cycle, we have a unique cyclin that's working with unique CDKs. For example, in G1, we have cyclin D working with CDK4 and CDK6. In our S phase, we have cyclin E working with CDK2 in the beginning of the S phase. However, early on in the S phase, our cyclin E is degraded and cyclin A takes over. So really, for the majority of the S phase, we have cyclin A CDK2. However, don't forget that this cyclin E is incredibly important in getting this S phase started. And we'll discuss this in a second. Then in our G2 phase, we have cyclin A CDK1. And in our M phase, we have cyclin B and CDK1. So let's look at an example of how these cyclins and CDK work. Just one thing I do want to mention before is that CDKs are present constantly throughout the cycle. Okay, so we'll always have CDK4, we'll always have CDK6, always have CDK1s, right? However, cyclins are only present in specific phases. We synthesize the cyclins at the beginning of each phase, and then by the end of the phase, we actually degrade them using our ubiquination and then eventually sending them to the proteasomes. So cyclins are short-lived, they only live for that particular phase, but our CDKs are present throughout. It's just that the CDKs will be activated in the presence of a certain cyclin. So let's look at an example specifically of our S phase with cyclin E and CDK2. Remember that we will have to synthesize the cyclin E in order for us to activate our CDKs, right? So, there might be certain triggers, like certain growth factors that want to push for growth and push the cell through the cycle, right? So they're going to help us synthesize these cyclins at the beginning of our S phase, or we can say in the end. Towards the end of the G1, we make a lot of cyclin E, okay? We make a lot of cyclin E so we can, use the cyclin to turn on our cyclin-dependent kinases or CDK2 specifically in this example. Once our CDK binds to the cyclin, it turns on, okay? And really, the cyclins don't do anything themselves. They simply regulate the CDKs. And then it's the CDKs, either kinases, right? So they're going to go and phosphorylate our target. And phosphorylation of the target will help us synthesize any proteins that we need to continue on in the S phase. For example, our helicases, topoisomerases, the whole list of proteins that we need to carry on our S phase. So this is how our cyclin CDK work. The cyclin regulates the CDKs and the CDKs phosphorylate our targets to keep this cycle going. So the next thing we want to get into is who's regulating the cycle. Remember that we can't be just growing all the time, right? Our cells, sometimes the cells get old, sometimes there's DNA damage and we need to repair stuff. So we can't just let this happen all the time. We need to have certain checkpoints and certain things that are regulating the cycle. And there's 2 things that we're going to be discussing. The first thing we'll look at is a retinoblastoma or RB. And RB acts as a checkpoint specifically at the G1 and S phase, and also at the G2M phase. But if you ask me, the G1S phase is really the most important phase, right? And it should make sense because this is really in early on in the cell cycle, when the cell needs to make that decision of whether it wants to invest all that energy and proceed into the S phase and the G2 and the M, right? So this is where you have to make that decision of whether you want to continue on into the cell cycle and keep dividing, or if you want to fix something, send it for repair, or anything of that sort. So the RB is working at both of these checkpoints, and we'll look at it in a little bit more detail. But first, let's talk about the other thing that's controlling the cell cycle. So the other thing that's really controlling the cell cycle is called P53. It's called the guardian of the genome. Okay, P53 can detect any kind of stress on the cell. Okay, and most specifically, it can send the cell. It can either send the cell to repair. So if let's say it finds some kind of DNA damage and it wants to repair it, it can also send the cell for apoptosis. And it can also age the cell. So it can also send the cell for senescence. Oh, cannot spell senescence. Senescence. Okay, there we go. Aging. How does P53 do that, right? So if it wants to repair the cell cycle, it's going to turn on another P protein called P21. P21 is present pretty much throughout the cell cycle, and it can block our cyclin CDK complexes in our G1, S, and G2 phase. Okay, so P53 by turning on P21 can cause cell cycle arrest in any one of these phases. P53 can also turn on our pro-apoptosis genes if it wants to induce apoptosis. Okay, so that's so let's look at RB and P53 in a little bit more detail. So let's look at RB. Remember that RB is the checkpoint. If RB is present and activated, like in this picture here, it's binding to E2F, which is a transcription factor, and it's stopping the transcription from taking place. Okay, if RB is active, E2F is inactive. If everything looks good though, and we want to proceed with the cell cycle, we have to inactivate RB. And how do we do that? To inactivate RB, we have to phosphorylate it. And we add phosphates to RB to shut it down. This allows us to activate our E2F, and the transcription factor is activated, and it can go on to transcribe the things we need for proliferation and growth and continue on with the cell cycle. I do want to talk about a small thing here and how RB, how we can, chemical modification for RB can actually lead to cancer. And to understand that, sometimes RB can be hyperphosphorylated. Okay, so when RB is normally phosphorylated, it can keep switching back and forth from the activated to the inactivated stage, okay? However, if we hyperphosphorylate RB, it is irreversibly inactivated. So it's irreversibly inactivated. What that tells you about E2F, that E2F will be constantly activated, and we've essentially lost our checkpoint, right? So we've essentially lost our checkpoint. We're having transcription of growth promoting genes all the time, and this can lead to cancer. When we lose a checkpoint, this can lead to cancer. Specifically, a hyperphosphorylated RB leads to retinoblastoma, which is usually seen in young kids, like 2 to 5 years of age, and osteosarcoma, which is something we see in kids in, let's say, 15 to 20 years of age more commonly. So that's the cancer kind of correlate that you want to remember about retinoblastoma gene. Next, let's look at P53, or the guardian of the genome. P53, if there's no stress in the cell, we don't need it, right? P53 is inactivated, and there's this thing called MDM2 that ubiquinates the P53, and it can send it for degradation. However, if there is stress in the cell, P53 needs to be activated, and we need to inactivate this MDM2. Okay, so in the presence of cell stress, for an example, if there's any DNA damage, we're able to turn on these specific genes called ATM and ATR that detect that DNA damage, and they're able to inhibit or turn off my MDM2. What this does is it turns on our P53 by phosphorylation. And remember, once P53 is turned on, it can either turn on pro-apoptotic genes, it can turn on our P21, right? And it can cause cell cycle arrest or send the cell into apoptosis. So that's how P53 works. Okay, so let's come back to the cell cycle. So we've discussed what makes the cell cycle keep going. We've discussed two things that act as checkpoints and stop the cell cycle. One thing I do want to mention here about P53 is that if we have a mutated P53, it is disastrous, really. Almost 50% of the cancers are because of a mutated P53, right? Because P53 is guardian of the genome, and if you lose the guardian of the genome, that's bad news, right? There's also a syndrome called the Li-Fraumeni syndrome that's associated with P53, in which people get multiple different cancers. So you have brain cancer, adrenal cancer, leukemias, the list just goes on. Okay, next let's talk about a specific P protein that works specifically in the G1 phase. There's this P protein called P16 or INK4A, and this specifically blocks our cyclin D CDK4/6 complex. And if this guy here is mutated, if it's a familial mutation, we see melanomas in the the patient, and if it's sporadic mutation of P16, then we mostly see pancreatic cancers. Okay, so that's P16. And I think that covers pretty much all our basic cell cycle inhibitors. The last thing I want to talk about here is in the M phase, our cyclin B CDK1 complex. Now this complex is very special, and there's a lot of control or things kind of inhibiting and activating this complex. So let's talk about this. The cyclin B CDK1 complex is together called the MPF, or the maturation promoting factor. Okay. The MPF interacts with three different things. So the first one, let's look at CDC25. This is a phosphatase. Okay. CDC25 is like a best friend. Okay, because MPF is able to turn it on, and it turns MPF back on and keeps our cyclin B CDK1 levels high and functioning. So these are like best friends. They both share the love. Okay. Then we have APC, which is called an anaphase promoting complex. This is like a bad friend, and we'll discuss why it's a bad friend at the end. But let's first talk about how APC works. So MPF is able to activate APC. Okay. Once APC is activated, it's able to turn off securin. So securin is a protein that turns on our cohesin protein, and keeps. Sorry about that. So it's a protein that turns on our cohesin protein and keeps my sister chromatids attached to each other. However, when we're in our anaphase, we need to break that cohesion protein and split our sister chromatids apart. So for that, we have to turn off securin, and that's what APC does. APC is able to take securin and send it for proteasomal degradation. It ubiquinates it and sends it for degradation. Once securin is turned off, this thing called separase is turned on. And separase can now go and separate my sister chromatids. Okay, so cohesin will be turned off, and separase will now be able to go and pull my sister chromatids apart to continue through my anaphase and finish my mitosis phase. So that's what APC does. Okay, it turns off securin and turns on separase in the process. Now APC, we've already seen that it's able to ubiquinate things, right? So it not only ubiquinates securin, but it also ubiquinates our MPF, and it sends their MPF or cyclin B-CDK1 complex for degradation. So that's why we said APC is kind of like a bad friend. It takes the love, but it doesn't send the love back. And the last thing we want to really quickly mention is WEE1, which is a kinase. And this is like an enemy. So this is not a friend. MPF doesn't like it, and WEE1 doesn't like it. They both inhibit each other. And WEE1 can also send MPF for degradation. So that's how our cyclin B-CDK1 complex is regulated and helps us get through the M phase or the mitosis phase. So this was like a really quick and dirty overview of cell cycle control, and I hope this was helpful. Thank you for your time.